Myself, Mihir A Hansalia. I am a research scholar in MSc at Anand Agriculture University, Anand from Gujarat. So basically, my research work is also on finger millet, and I choose this topic because this year is the International Year of Millets, and my topic is revolutionizing finger millet agriculture by breeding and biotechnological breakthroughs. So, for ease of understanding, I divided my seminar into six parts. First is the introduction. First is the introduction. In this part, I will cover botany, flower biology, nutritional importance, origin and evolution. And in the second part, I will talk about finger millet breeding, history, where I talk about history, breeding targets for improvement, wild relatives of wild relatives of the finger millet, then breeding methods. Third part is the case studies, where I will talk about the different aspects of improvement in the finger millet. Fourth is the conclusion. Fifth is the future thrust, and sixth is the achievement by breeding and biotechnological improvement through. First is the introduction. So finger millet comes under the kingdom Plantae, these are angiosperms, class monocots and order Poales, and family is the Poaceae, and the genus of the finger millet is Illucine. The botanical name of the finger millet is Illucine coracana, and the center of origin is East Africa. The chromosome number of the finger millet is 2n is equal to 36, and it is a allotetraploids. And the mode of pollination of the finger millet is self-pollination, and the outcrossing percentage is less than 1%. If we talk about the botany of the finger millet, then it is a highly self-pollinated, robust and annual growing crop. Stems are slender, glabrous, and capable of producing many tillers and nodal branches. As you can see in the figure 2a, the stem of the finger millet, and the finger millet inflorescence is known as ear, which is made up of 2 to 11 spikes. It resembles fingers on hands, hence its common name is finger millet. As you can see in the figure 2b, the inflorescence of the finger millet, where lowermost spike of the inflorescence is known as thumb and other spikes known as fingers. This is the picture of the immature inflorescence, and the figure 2c indicates the mature inflorescence, and it is a 30 to 150 centimeter tall and takes 3 to 6 months to complete the seed cycle. The grain is globose and smooth, and the color can be white, light brown, copper brown, and dark brown. In the figure 2d, you can see the seeds of the finger millet. Now we will talk about the floral biology of the finger millet. So florets are hermaphrodites having boat-shaped lemma, small palea with two loducules, and the terminal floret is sterile. Here you can see in the picture A. If we talk about the parts of androecium, then it contains three stamens. You can see in the figure B, where anthers are oblong and big, and the filaments are short. If we talk about the parts of the gynoecium, you can see in the figure C, it is two styles with plumostigma, and anthesis occurs between 2 to 5 am, and it takes 10 days to complete its flowering, and the pollen remains viable for 20 minutes while the receptivity of the stigma is up to 5 hours. This is the major concern in the finger millet because due to this the finger millet outcrossing is less than 1%. If we talk about the finger millet scenario of our world, India and Gujarat, then in the world, the finger millet cultivated into 28.1 lakh hectare, producing 38.3 lakh tons finger millet at a productivity rate of 1,362 kilogram per hectare, which contribute to 4% total millet production. In India, it covers the area of 11.1 lakh hectare, producing 17.6 lakh tons at a productivity rate of 1,585 kilogram per hectare, contributing 11% to total millet production in country. And if we talk about the Gujarat, then it is producing in 14,000 hectare area, and production is 13,000 ton, and productivity is 928 kilogram per hectare, which contributed 1% of total millet production. If we talk about the top five millet producing finger millet producing countries, then it is India, Ethiopia, Nepal, Uganda, and Malawi. And these five countries collectively producing 99.6% finger millet globally, whereas Ethiopia has the highest productivity level, 2,301 kilogram per hectare. If we talk about the area production and productivity of India, then 2013 to 2022, uh, area is declining over, till 2016, but after international national year of millets in 2018, it is increasing, and production and productivity is fluctuating over the years due to some area cultivated by other crops, and other reason is like using the traditional varieties of finger millet, like that, etc. If we talk about the area production and productivity of Gujarat, then production is highest in 2016, which is 27,000 hectare, whereas productivity is also highest in 2016, which is 1,400 kilogram per hectare. And the percent distribution of finger millet production in different states, then Karnataka, Tamil Nadu, and Uttarakhand contributed 88% of the production of the country, and other state contribute 12%. Then nutritional importance. We all know that these millets are known as nutri-cereal, where finger millet is the richest source of calcium, which is tenfold higher than rice and wheat. It is also rich in potassium, phosphorus, magnesium, vitamin A and B complexes, then 40% essential amino acids like isoleucine, lysine, cysteine, methionine. If we compare the millets, finger millet generally, and all other millets to the major cereals like rice and wheat, then finger millet is rich in carbohydrate compared to all other millets. It is also rich in fiber compared to major cereals like rice and wheat. And if we say the glycine index, which is a ratio of carbohydrate divided by fiber, then its ratio is very low compared to the major cereals rice and wheat. And the fat percent is also very low compared to the major cereals like wheat. And the major calcium and potassium is highest in finger millet compared to all other millets and major cereals. Then in the botany part, I describe about the different grain colors of the finger millet, which is white, brown, dark brown, copper brown. So, conducted study by Rao in 1994, it suggests that the white ragi is rich in protein, iron, and calcium compared to brown ragi, and it almost nil in white ragi. Tannin is nil in white ragi compared to brown ragi. This tannin contains increase the colors of the products, value-added products of the developed by finger millet from finger millet. So this could be very low, and which is low in the white ragi. Then origin and evolution. So finger millet originate from East Africa, you all know. Then the two theories suggest of the evolution of the finger millet. The first theory suggests that it could be originated from the Illicin indica based on the cytological, biochemical, and molecular evidence. Illicin indica contributed one of the genome to the cultivated Illicin coracana genome. And in contrast, the second theory suggests that Illicin coracana and Illicin africana are genetically related, and it might have originated from africana through a process of natural selection and subsequent mutation. If we talk about the principal growth stages of the finger millet spike given by Mirza and his co-workers in 2017, then four principal growth stages S1, S2, S3, and S4, where S1 is the booting stage, where one-fourth of the inflorescences has emerged, but florets are not yet identifiable, and it is green in color. S2 is the anthesis stage. Here, 50% of the anthesis is done, and inflorescence is yellow in color due to emergence of the anthers. S3 is the grain filling stage. Here, grain development is started, but the grain is still immature, so it can be crushed between fingers. And the S4 is the ripening stage, where 50% spikelettes are ripened, and the grain is hard, so it cannot be divided by thumbnail. And the influence color is also dark brown, from yellow to dark brown. Then the finger millet breeding, the core part of my seminar. If we see the history of breeding efforts in the finger millet, then in early 90s, Dr. Leslie Coleman released first finger millet variety H22 in India in 1918. Uh, After till 1950, finger millet development is very less due to its self-pollinating nature. But after development of contact method of hybridization in 1951, the finger millet improvement get a boost. And also in late 1950, the project for implementation of regional research on cotton, oilseed, and millets, there is awareness in the improvement spread in all over India. Till this, the improvement is confined till the southern states of the India, like Tamil Nadu, Karnataka, Andhra Pradesh. Third is the 1960 to 1980s, where hybridization between two divergent cultivars, African germplasms and Indian cultivars, are done by Dr. C.H. Lakshmianna in 1964, which resulted in the development of INDAF cultivars. Also, EICRB, India Coordinated Millets Improvement Project during 1965 started. After in 1990s, major finger millet growing state developed location-specific varieties where superior recombinant inbred lands also developed. Then in 2000 to 2010, core and mini-core collection developed from the globalization and whole genome sequence, transcriptome sequence of finger millet genotype ML365 also done in 2017, and also two biofortified varieties were released. Then breeding targets for improvement. I divided breeding targets in four parts where most have traits, basic traits, then long-term traits, and quality and market-driven traits. Most have traits include optimum plant height, days to flowering, number of productive tillers, bold grain size, etc. Where basic traits include blast resistance, trigger, drought, lodging, and high fodder, nutrient digestibility. While long-term traits include breeding to prevent seed scattering, high seed vigor, herbicide tolerance, and quality and market-driven traits, where breeding for grains rich in micronutrients, grain color should be white, which is high in puffing percentage, and taste. If we talk about the wild species of the finger millet, then it is Africana, Flocifolia, Indica, Intermedia, Jageri, Kaigensiensis, Multiflora, Compressa, and Tristacea, which is divided into three gene pool. Primary gene pool is the Elysian Korakan Africa, which is consist cultivated and wild species. Secondary is the Elysian Indicana, Tristacea, and Flocifolia. Third is the Jaegeri, Multiflora, Compressa, Kaigensiensis, and Intermedia. Figure 6 shows the different wild species of the Elysian genus, and plastid phylogeny is reported that Elysian africana, Korakan, and Kaigensiensis found to share a common ancestor with Elysian indica group, and biochemical and genetic evidence indicate a sister relationship between Elysian indica and Elysian tristacea. Breeding methods of the finger millet are pure line selection, mass selection, hybridization, and mutation breeding. These all are conventional breeding, and non-conventional approaches is like molecular breeding is done. Now, case studies. The first case study is on the development of transgenic finger millet resistant to leaf blast. The objective of this case study is introducing rice chitinase gene into finger millet using agrobacterium mediated transformation conducted by Ignacy Muthu and Caesar in 2012 in Chennai, India. Material is GPO45, where the rice chitinase gene inserted into the GPO45 by agrobacterium mediated transformation. Here, plasmid hygromycin chitinase 11 used. This plasmid contained rice chitinase gene, hygromycin phosphotransferase gene for the selection marker, and Gus gene for the reporter. Also, Gus assay, PCR analysis, southern blot, western blot are done to check the integration and expression of the gene. This is the protocol of the tissue culture protocol of the transform plants. Here, B shows the hygromycin resistant plants, transform plants. C shows the shoot induction. D shows the shoot elongation. E shows the root induction. And FG shows the hardening of the plant. And H shows the field establishment of the plant. Gus assay done for the check the expression in transform explants of the finger millet, where transformation of the finger millet highest seen in the uh, experiment number 3, which is 3.9%, and frequency of transient Gus expression seen in the experiment number 1, which is 19.3%. If then it Ignacy Muthu checked the PCR analysis, where in R0 and R1 progenies, amplification of Ki11 gene from genomic DNA isolated from R0 and R1 progenies shows the 0.584 kb band in both R0 and R1 progenies. Then, in the figure 11, it checked for the southern blot analysis for further confirmation of the integration of the chitinase gene, and it shows in both R0 and R1 progenies one copy of the in uh, uh, all 8 progenies of R0 and R1 plants. It shows the chitinase integration. Then Western blot analysis done for check the integration of the expression of the chitinase enzyme. Then R0 and R1 progenies both shows the 35 kilodalton protein in both R0 and R1 progenies plants. Then table 6 shows the chitinase activity in R0 progeny of finger millet plant transformed with rice chitinase gene. Then non-transform control shows the 5.8 chitinase activity, and transgenic line shows the 24.8 chitinase activity. Then lift blast assay was done, and this non-transform plants shows the disease resistant scale 8 and 9, where transgenic line shows the 0 to 4 disease resistant scales. This study demonstrated that the rice chitinase gene can be added in the finger millet through agrobacterium mediated transformation, which resistance give high resistance against leaf blast. We can add all other finger fungus gene, resistant fungus gene into the finger millet. This figure 13 shows the photograph of the control and transgenic plant. Here you can see in the leaf after spray of the spores of the fungus after 25 days control is susceptible and the transgenic is still resistance. Second is transcriptome analysis of finger millet reveals unique drought responsive genes conducted by Parvati and her co-workers in 2019 to identify the characterized unique drought responsive genes and associated pathways using transcriptome in finger millet. Plant material uses GPU28 and treatments given like sample A is the 100% field capacity and sample B contained pool of 80%, 60%, 40%, and 20% field capacity. Here, we estimate the cell viability to check the drought stress effect on the plants. Then it sees that uh, wherever higher drought stress, there is a reduction in cell viability. Then he sequenced the finger millet, and it shows that drought sample contains more transcript than control, which shows that drought sample have more gene expressed than control. Then they do the CAG analysis of the finger millet. Then it shows the total of 131 unique pathways belonging to 19 groups were identified under 5 major categories. And this pathway provide valuable resources for investigating specific processes, function, and pathways in plant drought stress research. Then they analysis DEG analysis to check the expression of the genes. Here in sample B, the green indicates higher expression and red indicates lower expression of the genes. In figure A, sample B shows the higher expression of these genes, where sample A shows the lower expression of these genes, and figure B, here sample B shows the lower expression of genes, and sample A shows the higher expression of genes. And case study 3, hybridization and hybrid detection through molecular markers in finger millet, conducted by Krishna and her co-workers in 2020. The objective is to compare efficiency of two different emasculation methods and to analyze the genetic purity, conducted at Payur, Tamil Nadu, India. Two parental lines I2606 and PR202 used. The morphological character described here, and total 149 primers, where 28 is RAPD, 20 is ISSR, and 101 is SSR was used, and two emasculation method, hot water treatment and hand emasculation used. Then result and discussion. If we check the conformation of F1 hybrid using RAPD primer, then some primers gives the bands in all the F1 hybrids like OPK10. That gives one specific band in male parent but no band in female parent. Then all bands come in F1 hybrid, but some primers not give the results. So they go further by ISSR marker ISM marker also gives the same, like somewhere it gives the bands in all the F1 habits, like in this UBC 857, where somewhere it not give reliable results. But in confirmation of F1 habit done by SSR markers, then in first cross done by where UGEP67 primer used, then it gave one specific band in male and one specific band in female, and both bands are comes in all the F1 hybrids. And this in same cross UGP22 primer used, then it gives the same result. And if we done in the reciprocal cross, then UGP67 gives one specific band in male and one specific band in female, and both bands are present in the all F1 hybrids. The same result given by UGP22 primer in same cross. And table 9 shows the evaluation of exclusion method and genetic purity of F1 hybrid of finger millet using SSR marker, where hot water based emasculation gives 80% purity, whereas hand emasculation gives 40% purity. So, this case study concluded that the SSR marker, which is codominant in nature, gives the reliable 100% accurate result in hybridity testing, whereas hot water is the most efficient method compared to hand emasculation, and genetic purity also highest in hot water method compared to the hand emasculation method. And molecular based detection of the hybrid testing, we can use it in a check the genetic purity compared to the GOT test. The fourth is the early identification of the salt tolerant genotype in finger millet germination stage by observing the morphological characters, conducted by Divya and her co-workers in 2022. The objective of this case study is to find out NaCl tolerant and NaCl sensitive cultivars of finger millet at the germination stage, conducted at TNU, Tamil Nadu, India. Materials and methods: the 15 different cultivars used and different NaCl salt concentration like 50, 100, 150, and 200 millimolar used. Method: after the treatment, 20 typical seedling tested for germination, shoot length, root length, fresh weight, dry weight, and vigor index. Table 10 shows the effect of the NSL stress on the germination percentage, where GPU 28, 67, ML365, Udrumaliji GPU 48, and PYR1 shows the 70% or more germination in 150 millimolar concentration, where only ML365 shows the 90% germination in 200 millimolar concentration, whereas VL400 and KM252 shows the lower number of the germination. Table 11 shows the effect of financial stress on shoot length, where same cultivars described earlier shows the higher mean shoot length, more than 1.10 centimeter compared to all other cultivars, whereas VL 400 and KM 252 shows the lower number of the shoot length in the experiment compared to all other cultivars, where table 12 shows the effect of axial stress on root length, where GPU 28, GPU 67, ML365, Udurumaliji, PYR1, and GPU 48. This all shows the root length more than 2 centimeter, where VL400 and KM252 shows the lower number of root length, where vigor index, same cultivar described earlier, shows the root length vigor index more than 220, and the VL400 and KM252 shows the bigger index lower than 60. Correlation: if we correlate, then germination percentage is highly and significant correlated with shoot length, whereas non-significant with the dry weight, and vigor index, uh, which is significant and positively correlated with all other traits. Then the case study 5: characterization of finger millet global germplasm diversity panel for grain nutrients content for utilization in biofortification breeding, conducted by Bakya Lakshmi and her co-workers in 2023 at Ikriset, Telangana, India. Objective of this case study is to find the finger millet accession for grain nutrient content, study the association of grain nutrient traits with agronomic traits among land-basis breeding land germplasm from Asia and Africa, and identify the finger millet accession with superior multiple grain nutrient content. Here, 314 finger millet germplasm used in alpha latex design with 3 replication for 2 years, 2018 and 19, which divided based on geographical representation, diversity based on race, and biological types. Result and discussion shows that Planaris is high grain yield, high calcium, and high potassium compared to all other race, where Vulgaris race is high in protein percentage, and It is early in the flowering. Table 16 shows the mean comparison of agronomic and grain nutrient traits in finger millet among regions and biological status, where African germplasm shows all traits good than Asian germplasm except iron, and it is late in flowering compared to Asiatic germplasm, where based on the biological status, breeding lines are high in grain yield and potassium, and land races is high in zinc and calcium, and breeding lines is early in the flowering than the land races. Table 17 shows the comparison of traits based on finger millet green color. Then light brown germplasm has high grain yield compared to white. Reddish brown germplasm has higher iron content than light brown. And if we divide the group two groups, the colored and the white, then white germplasm has the high copper and potassium than the colored one. Table 18 shows the correlation among grain nutrient and agronomic trait based on breeding lines and land races, where breeding lines has a protein which is a significant and positively correlated with iron, which is same in the land races, but in land races protein is significant and positively correlated with zinc, but it is non-significant associated with inbreeding lines. If we see the calcium, then it is a positive and significant correlated dish to 50% flowering in both breeding lines and land races, and calcium with grain yield is non-significant in both land races and breeding lines, and calcium with protein is negative and significant correlated in breeding lines, but in land races it non-significant association with protein. If we see the iron and comparison correlation with magnesium, copper, sulfur, and phosphorus in land races, then it is positive and highly significant, where it is non-significant in breeding lines. Now, based on region wise, then calcium is positive and significant correlated with h to 50% flowering in both Asiatic and African germplasm, whereas in African germplasm, calcium is positive and significant correlated with zinc, but non-significant with zinc in Asiatic germplasm. Grain yield is non-significant associated with magnesium and copper in African germplasm, whereas this same traits is significant and positively correlated in Asiatic germplasm. Zinc. Zinc is non-significant associated with copper, phosphorus, and potassium in Asiatic germplasm, whereas it is highly significant and positively correlated with copper, phosphorus, and potassium in the African germplasm. Based on this analysis, researchers divided some top 10 association based on the multiple grain nutrient content. Then 2872, 2875, this shows the highest zinc, calcium, and magnesium. Then Champlasm 817, 2008, 3935, 6473, 7386 shows the highest iron, zinc, and protein percentage. And Champlasm 3973, 5433, 2760 shows the magnesium and potassium highest. This all Champlasm are land races. So and all are coming from the five countries: India, Zambia, Uganda, Kenya, and Malawi. Conclusion: the first is the introduction of the rice chitinase gene through genetic modification enhance the plant's ability to resist pathogens by ensuring continuous and early expression of the defense protein, effectively preventing the establishment of harmful fungi. Second, essential cell function represented by TAFs were crucial for finger millet's ability to withstand drought. By recently discovered genes like PP2A, CIPK31, FPS, and SRPR alpha has the potential for drought response. Third, hot water emasculation was found to be more effective and convenient than hand emasculation, resulting in higher genetic purity of F1 hybrids. Whereas GPU 28, ML365, GPU 67, Udurumalije, PYR1, and GPU 48 demonstrated high salinity resistance, suggesting their potential for further crop breeding efforts to enhance salinity tolerance. Whereas diverse germplasm collection identified accession rich in grain nutrients, offering valuable candidates for biofortifier cultivar development in finger millet. Which is first, we have to develop climate smart finger millet varieties, then improve the nutritional content of finger millet, which is low in the finger millet like iron, zinc. Then to enhance the economic viability of finger millet by targeting genes associated with fodder improvement, bold grain size, increase green production, then to state the multiple beneficial traits in single finger millet variety to develop high performing crop. And the achievement, improved finger millet varieties released in India. Some varieties like Phule Kasari, Birsa Marua 3, Depole 3, Chhattisgarh Ragi 3, these all are high in the grain yield, more than 2 ton per hectare, whereas the some varieties like VL 382, Vegavati, Gujarat Nagli 8, KMR 340, these are the high in disease resistance. Then Gujarat Navsari Nagli 7, Navsari Nagli 6, and Gujarat Nagli 5 and 4. These are all are early mature, and Gujarat Navsari Nagli 7 is a white seeded variety. Now, biofortified varieties like CFME 1, 2, and 3. CFME 1 is the Indravati, released in 2020. And average grain is 3.11 ton hectare, which is multi-disease and multi-pest resistance, and calcium content 428.3 milligram per 100 gram. CFME2, GN9, and Gira, released in 2020, which is also registered to leaf blast, and calcium is 454 milligram per 100 gram. And CFME3 Equij, released in 2021, moderately resistant to leaf blast, finger blast, neck blast, and rich in calcium, which is 470 milligram per 100 gram. Now, I will conclude my seminar with one sentence that the rice eater is a weightless like a bird. The one who eats jowar is strong like a wolf, but one who eats ragi remains nirogi throughout his life. This taken from the old Kannada scripture. Thank you.
Thank you very much, sir, for a wonderful presentation. I will take up the questions from the participants now. In the meantime, can you please stop sharing the screen so that we can see your full video?
Okay. Just a minute now.
Okay. The first question is, how
Yes.
have recent biotech advancements contributed to enhancing the yield of finger millet crops?
Actually, ma'am, the recent biotech advancement like the QTL identification, then the transcriptive analysis gives the genes which is stress resistance. Like I mentioned in the my slides that the PP2A genes and the other QTL identified, which can be used in the biological advancement like genomic selection, then the CRISPR-Cas9 editing technology, which is the advanced technology in the biotech area. So we can use these techniques for improvement of the finger millet.
Okay. What specific breeding techniques are being employed to develop more resilient and high yielding finger millet variants?
Breeding methods generally used in conventional breeding is the pure line selection, then the mass selection and hybridization. But hybridization is a little much difficult because of the nature of the flower biology of the finger millet, because it is pollen is remain viable only for 20 minutes and the stigma receptivity is only for 5 hours. Due to this nature, the we can breed the varieties, but we have to be more precise. Then we can use the biotech methods as I mentioned in the first question's answer.
Okay. Can you explain the key genetic modifications that have been introduced to improve finger millet's resistance to pests and diseases?
Generally, some genes are identified, which is resistance to novel blast resistant genes identified in finger millet. But currently, I don't remember which paper I have read at that. But some genes are identified which resist the pest and disease also. Not pest, but only disease. The pest improvement is generally running in the improvement area.
Okay. In what ways do these breakthroughs address challenges such as climate change and changing agricultural landscapes?
Generally, if we see the climate change, then the finger millet is already climate resilient, that it can adopt in the any area like drought. If matlab means if it is a limited drought, then it can be adopted that condition. Then also in it is heat tolerance. Then it is also what says that if we have keep the grain of the finger millet, then it can be kept for we can kept it for more than decade. It not harmed by the pest attacks, but it is a limited. So we have to develop that type of varieties.
Okay. How are scientists ensuring that the nutritional content of finger millets is maintained or improved through biotechnological inventions?
Yes.
You got my question, sir? Hello.
Hello.
You got my questions, Mr. Mihir?
No, can you please repeat it?
Yeah, I will repeat it. How are scientists ensuring that the nutritional content of finger millet is being maintained or improved through biotechnological interventions?
No, I don't get it the question.
Okay, I'll take up the next one. Are
Okay.
there collaborative efforts between researchers and farmers to implement these advancements in real world agricultural practices?
Not this practices, but a participatory breeding is running in the finger millet, where some of the in southern states like Tamil Nadu, the companies do the participatory work with the farmers.
Okay. What role do precision farming techniques play in optimizing the benefits of biotech breakthroughs in finger millet agriculture?
No idea on this question.
Okay, okay. I'll take up the next question. Our policymakers supporting the adoption of these technologies and what challenges do they face in doing so?
If we develop the like a transgenic finger millet, then we have to take some norms, regulation like genetic improvement approval committee. We have to take some rules and regulation. Under that, we have to develop the varieties. So we have to take that type of the concerns in mind. Then we have to develop the variety.
Okay, Mr. Mihir, just see if you can answer this question. Can you highlight success stories where local communities have experienced positive outcomes from adapting biotech enhanced finger millet varieties?
Actually, there is no scope of the biotech developed variety in this time, but actually, uh, this need to work in the biotech area of the finger millet when we have to develop the varieties, because there is no such success stories like biotech improve finger millet varieties, because I say that it is still the orphan crop and we have to uh, discover that crop by biotech throughs.
Okay. What ethical considerations are taken into account in the development and deployment of technological solutions in finger millet agriculture?
No idea actually.
Okay. 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 Can you tell us about yourself? Few lines about yourself.
About myself,
Yeah.
actually I am a student, MSc student, from pursuing my masters in the genetics and plant breeding in the BA College of Agriculture, Anand Agriculture University, Anand, Gujarat. And my research is also on the finger millet, and my research is like evaluation of the germplasm, different germplasm for its morphological traits. Then I have also research objective on the molecular diversity, where I have to identify the diverse germplasm within this area of finger millet, and also nutraceutical profiling is I also uh, currently my master pursuing. So this is myself.
Okay. How did you get interested in this field?
What, ma'am?
How did you get interested in this particular field?
Actually, I have also background of the farming.
Ah,
My grandfather is also a farmer, and my father is in the value-added businessman in the value addition of the agriculture products. Yeah, so I also get the idea to join this field.
okay, that's very interesting. Thank you very much. We have now completed the round of questions. On behalf of agricultureinformation.com, we like to thank Mr. Mihir A. Hansalia for the talk
Thank
and for answering the questions. I like to also thank all the participants in the meeting. This meeting will now be closed.
Okay, thank you, ma'am.
You're most welcome.